Hello friends, in this session we'll be dealing with microbiology and the most important topic, that is sterilization and disinfection. All of you know that microorganisms are ubiquitous. In order to prevent contamination or decay by microorganisms, we need to remove them from surfaces. In this regard, the topic sterilization and disinfection is very important. We'll see some definitions. What is sterilization? Sterilization is a process by which an article, surface, or media is made free of microorganisms, either in the vegetative or spore form. So it's a complete term, that is, we are removing the burden of live microorganisms. It's a complete removal. Coming to the next term, that is disinfection. The sterilization and disinfection go side by side. Disinfection is the process of destruction of pathogenic microorganisms capable of giving rise to infection. And there is also another term, antisepsis or antiseptics, which we commonly hear. That is, these are chemical disinfectants which are used to prevent infection by inhibiting the growth of microorganisms. And this term is usually applied safely to living tissues. Now, there are certain terms which need to understand in the process of sterilization, that is bactericidal agents. Bactericidal agents are agents which are able to kill bacteria, whereas bacteriostatic agents are agents which inhibit the bacteria. And that is, they prevent the multiplication of bacteria, but most of them remain alive. Now, coming to the sterilization uses, that is, it's in the microbiology laboratory, sterilization is used to removing microorganisms from media as well as reagents. And also in food and drug manufacturing industries, it ensures safety from contaminating microorganisms. And in a hospital setting, it's also very, very important. That is, it's required for sterilizing materials, instruments, that is surgical procedures, diagnostic procedures, and operation theaters in order to control the spread of cross infections. Methods of sterilization. Two methods, physical and chemical. We look into the details of these two methods. Physical methods of sterilization. Physical methods, we can list 6 under them, that is sunlight, drying, heat, filtration, radiation, ultrasonic and sonic vibrations. And under the heat, we have 2 types of heat, that is dry heat and moist heat. And among all these, heat is one of the most important method of sterilization. Physical methods of sterilization. Sunlight. Sunlight, we know that it has got an appreciable bactericidal activity. And the ultraviolet rays in sunlight cause the killing of bacteria or microorganisms. And it is a natural method of sterilization of water in natural water bodies such as lakes, rivers, tanks, etc. Next one is drying. As we all know, the moisture is essential for the growth of bacteria. 3/4 of the bacterial cell is constituted by water, and hence drying has got a deleterious effect on bacteria. But the spores are unaffected, so hence it is an unreliable method for sterilization. Coming to the third one, the most reliable method, that is heat. Actually, there are 2 factors influencing sterilization with heat. The 2 types of heat, that is, one is dry heat and moist heat. Nature of heat, that is dry heat and moist heat. And next one is temperature and time. Time, actually, it's inversely proportional to temperature. Lesser the time or lesser the temperature, more the time required. Whereas higher the temperature, less time required. Regarding heat sterilization, we have to look into these terms, that is thermal dead time, decimal reduction time, and F value. That is, thermal dead time is the shortest time needed to kill the microorganisms in a microbial suspension at a specific temperature and under defined conditions. That is, if you are keeping all the others, oxygen, pH, carbon dioxide content constant, and using a specific temperature, say for 50 degrees centigrade, we have to see what is the shortest time needed to kill all the organisms in the microbial suspension. Next one is decimal reduction time. Decimal reduction time means you can say that, understand that it is a fraction, or it's also known as D value. The decimal reduction time is the time required to kill 90% of the microorganisms or spores in a sample at a specified temperature. Decimal reduction time, or D value, is the time required to kill 90% of the microorganisms or spores in a sample at a specified temperature. Third one is F value. F value means it's a time in minutes at a specific temperature. That is, usually it is 250 degrees Fahrenheit or 121 degrees Celsius, which is needed to kill a population of cells or spores. So that is F value. Actually, these values are very important in food industry, food manufacturing, as well as drug manufacturing industry. Mostly it's important in the food industry. Next is, we've already seen that factors influencing heat sterilization. One was the nature of heat, and other one was the time, then temperature and time. Third one is the number of organisms present, spores, etc. And the vegetative forms, all of you know that vegetative forms are very vulnerable to heat. Vegetative forms are killed by moist heat at 65 degrees centigrade and 30 minutes. And whereas the spore forms are the most resistant ones, they need a higher temperature. Higher temperature means lesser time. So 121 degrees centigrade for 15 minutes. Next one is the characteristic of the organism. What type of organism we have to kill, whether it is a sturdy one or a delicate one, and such as species, which species of microorganisms such as Mycobacterium, which causes tuberculosis, is very sturdy that it cannot be easily killed. Corynebacterium or cell deficient forms, they may not be easily killed. So that is characteristic of organism. Whether it is a spore-bearing ones will not be easily killed. So that is the characteristic of the organism, such as species, strain, sporing, etc. Then next, the type of material, whether the presence of organic material or substances require more time to post sterilization, such as organic protein content or smooth sterilization time will be more, etc. So that is the different factors which affect the sterilization. So we have seen the definition, types of sterilization, methods of sterilization, the different types, and we have seen the physical method of sterilization. The physical 6 points are there. Thank you.